We're going to launch into really the nerdiest and most boring part of this, the joint anatomy and physiology. Yeah, it's just sort of a chance for me to refresh your memory about how joints are put together and some basic physiology. After that, I'm going to discuss the pathophysiology of the joints with particular emphasis on arthritis and tendinopathy. Again, it's a lot of information. It's going to come at you very fast. Hopefully, most of you have encountered at least a large part of this information at some point in your careers. So, what is a joint? A joint is quite simply a structure where two bones come together and are connected in some fashion. Two bones come together and are connected in some fashion, which links the entire skeleton into an integrated and functional system. So, for example, the connection between your mandible and your skull is a joint, the connection between your tibia and your femur is a joint, but the connection between your gastroc and your calcaneus is not a joint. It's just where two bones come together. The joint may be mobile or it may be immobile. So, for example, your skull has a bunch of joints in it which are hopefully immobile, right? The joint between your ilium and your sacrum is supposed to be relatively immobile. The knee joint is supposed to be relatively mobile. There are a bunch of ways to classify joints, and we're going to look at a few of them. The first one is the structural classification, which differentiates on the basis of the binding tissue. So in other words, what holds the two joints together. For example, you can have primary cartilaginous joints or the synchondrosis, in which the binding material is either hyaline cartilage or fibrocartilage. And you see this in sutures and in the growth plates of long bones. In a secondary cartilaginous joint like the symphysis, you have hyaline cartilage and fibrocartilage. In the fibrous joint, like the syndesmosis of the radial-ulnar joint, you have tough elastic tissue. The gomphosis is the special name for the joint between your tooth and your mandible or your maxilla. And then synovial joints, which are the focus of our discussion today, have an intervening synovial cavity and a joint capsule which is filled with synovial fluid, and there's generally cartilage and or fibrocartilage as well. And we'll discuss all of that in detail. A functional classification differentiates joints on the basis of their movement pattern. So a synarthrosis has no movement, an amphiarthrosis has slight movement, and the diarthrosis, which is the joints that most concern us most of the time, are freely movable joints, and they tend to be synovial joints. And they are further subclassified on the basis of how they move: plane, ball and socket, hinge joints, pivot joints, condyloid joints, and saddle joints. The biomechanical classification differentiates on the basis of anatomical and biophysical properties. So, a simple joint is simply two articular surfaces in contact. So that would be like your hip joint, right? Or your glenohumeral joint, a simple ball and socket synovial joint. A compound joint contains three or more articular surfaces, and a good example of this would be your knee, which is not only the tibiofemoral joint, but also the patellofemoral joint. So the knee is a compound joint. The knee is also a complex joint because in addition to the two articular surfaces, or in the case of the knee, three, you also have a fibrocartilaginous component to the joint, and that is the meniscus. Right? So the knee is both a complex and a compound joint. Our focus today, to the extent that we focus on individual joints at all, will be on the knee, shoulder, and hip. And if you think about it, these are three very relevant joints to us as starting string coaches. We'll discuss a little tiny bit about the anatomy of these joints, but mostly that will be sort of a self-study program for you. Our focus today is on the synovial joint. We're not going to talk about, for example, SI joints. We're not going to talk about the spine. We're going to focus on these big synovial joints that most affect the way that we train and the way that we coach. A synovial joint is simply a cartilaginous joint which is enclosed in this fibrous capsule. Two bones come together, and where they come together is enclosed in this fibrous capsule, like a big, kind of gasket filled with fluid. All seems very mechanical and engineering. And the joint capsule, that big gasket, is itself a complex structure. which has an outer fibrous component, extremely tough, continuous with the periosteum of the bone, and an inner layer, which is a cellular diverse layer that carries out multiple functions. That's called the synovial membrane. It produces the synovial fluid, and it also keeps the synovial fluid clean, and it is subject to various immune and inflammatory stimuli. The two ends of the bones in this diarthrosis, this synovial joint, are covered by cartilage, and there are different types of cartilage, as we'll see, and the type that we find in synovial joints is hyaline cartilage, or what is simply called articular cartilage. And the synovial joint may also incorporate a fibrocartilaginous component like the meniscus. Now, when we talk about cartilage and synovium, what we're talking about is connective tissue. And connective tissue is kind of our thing as coaches. And cartilage, tendon, and bone are all connective tissues, and all connective tissues have certain things in common. Right? So they all have the same embryonic origin. They're all of mesenchymal origin. And the way that they're structured, even though they seem very diverse, is very similar. So all connective tissues consist of cells, living components, embedded in a non-living extracellular matrix, which incorporates minerals, particular kinds of polysaccharides, and certain types of protein. So if you look at any connective tissue, you'll find a cell type, a matrix type, and a protein type. So for example, with bone, the living elements are the osteoblasts and the osteoclasts. The osteoblasts and osteoclasts. And in all of these connective tissues, you'll find a cell type that lays down the non-living matrix and a cell type that breaks down the non-living matrix, so that there's this potential for constant remodeling of the matrix. So in bone, the cells are the osteoblasts and the osteoclasts. The matrix is a mineralized matrix, largely hydroxyapatite, this calcific mineral matrix of the bone. And the protein is this sort of collagen substructure or collagen template for the bone. If you look at cartilage, the living components are the chondroblasts and the chondrocytes which lay down the cartilage matrix, the chondroblasts which break down the cartilage matrix, and the matrix is primarily this non-living substance, chondroitin sulfate, which we'll discuss, and the protein component is collagen and elastin of varying proportions depending on the cartilage type. In tendon, we find tenocytes and tenoblasts, or fibroblasts, which respectively lay down and break down the tendon matrix. The matrix is composed of proteoglycans, polysaccharides, glycosaminoglycans, and so forth. The proteins are collagen and elastin, again in varying proportions. And if you look at another connective tissue, blood, you find the same pattern. You wouldn't think it is, but it is the same pattern. You have living cells, erythrocytes, leukocytes, lymphocytes, platelets, embedded in a non-living matrix, the serum, right? Which is filled with specific proteins, albumins, complements, immunoglobulins, clotting factors, and so on. So all of these connective tissues really have this sort of same underlying structure in a certain way. When we look at cartilage in particular, we find that we're dealing with a variety of different types of tissues. Cartilage is not just one tissue, but all forms of cartilage are formed by chondrocytes embedded in this extracellular matrix, which is dominated by collagen of different types, proteoglycans, elastin, and other non-living substances, particularly chondroitin sulfate, which we'll discuss in a moment. Cartilage is highly avascular and highly aneural, which means that it does not have a dedicated blood supply. There is no capillary bed in the cartilage, and you wouldn't want one, right? And it's highly aneural. It doesn't have dedicated sensory nerves, and you wouldn't want those either, right? So it's largely non-living material with living elements embedded in it. It's terribly atrophic. It's a living tissue, but its turnover is extremely small. And its adaptive capacity is temporally very limited. It does not regenerate to any clinically significant degree, and this will be an ongoing theme, during this discussion, it does not hypertrophy. It doesn't get swollen, it doesn't get jacked, it doesn't have gains, right? It can't respond to loading in the way that some other tissues like muscle tissue can. It receives its nutrients from the synovial fluid, primarily by diffusion. That includes its oxygen and its glucose supply. So the synovial fluid is constantly pumped through the cartilage matrix in synovial joints. The forms of cartilage, I don't want to belabor this too much. You have elastic cartilage found primarily in the ear and the epiglottis, the cartilaginous trapdoor that covers your larynx, and it's primarily dominated by elastin. Fibrocartilage is what you find in the menisci and the intervertebral discs of the spine, and it's more type 1 collagen and a lot of fibrous tissue as well. It's extremely tough. It's found in the symphysis and in the intervertebral discs. And again in menisci. The kind of cartilage that we're going to be talking about most today is hyaline cartilage, which is just called articular cartilage or joint cartilage. And the matrix in hyaline cartilage is chondroitin sulfate. Now, what is this chondroitin sulfate, besides something that you pay too much for at Walgreens? Chondroitin sulfate is a polysaccharide. It's a polymer, and the monomeric unit of the polymer, the unit that gets strung together to form this polymer, is itself a disaccharide, right? A disaccharide formed of N-acetylgalactosamine and glucuronic acid. N-acetylgalactosamine is important because it is a nitrogen-containing sugar, and that will be something that we'll touch on in a moment here. If you look at this disaccharide monomer of the chondroitin sulfate, you'll see a lot of these little R's that are hanging off of it here. Well, R is not an atom, right? There is no element R, right? R indicates that that's where you can stick an R group, a variable R group. And in the case of chondroitin sulfate, what you're talking about is sulfate, right? The sulfate group hangs off this sugar. This polysaccharide has a lot of sulfate groups hanging off of it. Why is that important? Because sulfate is basically a group consisting of a sulfur atom surrounded by a bunch of oxygen atoms, and it has a lot of electron density around it, meaning it has a lot of negative charge. So this sugar chain is coated in negative charge, right? And negative charges repel each other. So these chondroitin sulfate chains repel each other. There's a lot of electrostatic repulsion between them. And what that means is that chondroitin sulfate as a substance is highly resistant to compression. The electrostatic forces within the matrix resist compression greatly, which is part of what gives cartilage its incredibly tough properties. It deforms very little during loading, and CJ will talk about that in depth in his section. Now, this N-acetylgalactosamine is a nitrogen-containing sugar, right? And all nitrogen-containing sugars in the body are derived from or synthesized from a common precursor, glucosamine, which is also something that you pay too much for at Walgreens when you buy your joint supplement. And that is part of the rationale for including glucosamine in joint supplements. All the N-acetylgalactosamine in the chondroitin sulfate is derived from glucosamine. Therefore, you should eat glucosamine because it will be magically transformed into more cartilage in your joints, and you'll be supple like a leopard, right? So that's the theory. And once again, you can see how all these three different forms of cartilage consist of the cute little cells there embedded in this non-living matrix, right? With elastic cartilage being more cellular, fibrous cartilage being almost acellular, and hyaline cartilage somewhere in between. Once again, here in this picture, you see the chondrocytes embedded in the matrix. These are very long-lived cells that synthesize the cartilage matrix, and they are subject to mechanotransductive signaling. They do respond to loading, right? They just don't get in a real hurry about it, and they're not real super motivated about it, but they do respond to mechanotransductive signaling. There is potential for remodeling of cartilage at the molecular level, right? Although not at the level of cartilage hypertrophy. Again, this is something that we're going to touch on over and over again. In the joint tissues, the cartilage and the tendon and the ligament, and to a lesser extent the bone, there's very, very little potential for hypertrophy. There's what we call an uncoupling between the loading and the mass adaptive response, right? And if you think about it, it kind of has to be that way. I mean, you don't want your cartilage really to hypertrophy. You don't want it to wear out, but you don't want it to get huge and get swole either, because that would deform the mechanical characteristics of the joint, so that would lead to its own problems. The synovium is this inner lining of the joint capsule. It's a specialized connective tissue, and it forms this membrane inside the joint capsule gasket. And unlike the cartilage, it's heavily vascular. It's got an extremely rich blood supply and an extremely rich nerve supply as well. It's quite sensitive. The inner layer is composed of two different cell types, these type A cells, which are macrophages, which keep the synovial fluid clean and pristine and free of bugs and so forth, and which will respond to immune and pro-inflammatory stimuli, as we'll see later, and type B cells, which are modified fibroblasts that actually produce the synovial fluid. The synovial fluid itself is kind of like urine. In a way, right? It's an ultrafiltrate of plasma produced by the synovial cells. It has a number of unique physical properties. It's what we call a non-Newtonian fluid, or what some people would call a non-Newtonian fluid. I don't know anything about fluid dynamics, right? But basically what this means is it has these unique rheological or rheopectic properties. It responds to stress. So synovial fluid gets thicker and more viscous under loading, and it gets thinner and more watery, if you will, under unloading. So it actually responds to stress, at least in the young, healthy joint. In older, less supple leopard joints, it doesn't respond quite as well. It covers every articular surface within the synovial, within the joint capsule. It fills the entire joint capsule. And it actually circulates through the cartilage material itself. And this is the idea of weeping lubrication. So as the cartilage is loaded, it kind of releases more synovial fluid and other lubricating components into the joint space. It contains glucose and oxygen and electrolytes from the plasma. It contains hyaluronin, which is a mucopolysaccharide-like substance that lubricates the joint further. And this synovial fluid, by the way, this hyaluronin, is kind of like, almost like sort of a liquid cartilage. It's also composed of glucuronic acid and N-acetylglucosamine, and various other lubricating substances like lubricin, and a certain number of proteases and collagenases, which are exactly what they sound like, enzymes that will actually break down protein and collagen. There may also be growth factors in the synovial fluid, although I couldn't find as much about that as I would have liked. Again, with aging, the synovial fluid's rheopectic properties gradually decline, and it becomes less efficient, probably having something to do with the molecular structure of the hyaluronic acid as we get older. So, that's the joint capsule, and that's the cartilage. Now we're going to turn our attention to the other big component, tendon and ligament, with our real focus being on tendon, because tendon is the more dynamic of the two tissues, right? These are viscoelastic fibrous structures. What does that mean? It means that they're springs. They're dynamic. They can store and release energy in a cyclic manner. So if you have in your head this idea that tendons and ligaments are these sort of like ropes or cables, get rid of it. They're not, right? They are living structures with very profound viscoelastic properties, particularly the tendon. Once again, they're just a connective tissue. What do all connective tissue have, right? They have living cellular components embedded in and producing a non-living extracellular matrix with a proteinaceous component. In tendons, we have the tenocytes and the tenoblasts, or fibroblasts, which produce the collagen fibers and also break down the collagen fibers, right? The collagen fibers are arranged into fascicles. The fascicles are arranged into bigger fascicles, and the fascicles are arranged into a complete tendon. So this is very reminiscent of the way that muscle is organized, right? This incredibly hierarchical, beautiful structure, a bundle of bundles is what I always like to call it, just like muscle. At different levels of organization, these fascicles are wrapped up in little fibrous sheaths called endotenons, and the entire tendon is encased in a epitenon, another fibrous sheath that encloses the entire tendon. The entire tendon may be further enclosed in sort of a loose, almost sort of areolar sheath called a peritenon, which kind of insulates it from the surrounding tissue, or it may be encased in a synovial tendon sheath, right, with a synovial membrane filled with synovial fluid, rather like the joint capsule, right? A sheath that is filled with synovial fluid, particularly in like the fingers and toes, right, where the tendon has to go over sort of a little hook or pulley and allows you to do that. So you need something that will basically protect the tendon from abrasion. That's what the synovial sheath does. It allows the tendon to slide in and out. Probably shouldn't be doing that. Slide in and out smoothly, right? So, moving forward. So this is the tendon and ligament. Again, it's a bundle of bundles, right? A bundle of bundles, a highly hierarchical structure. That's the gross structure of the tendon. At the ultrastructural level, what we have are these collagen fibers, right? Collagen is basically made up of these tropocollagen monomers, which come together and form these collagen fibers, which are then arranged in a staggered array to form the collagen fibers of the tendon. And these collagen fibers are heavily cross-linked to each other. So what you end up with is this sort of coaxial structure, which is incredibly tough and incredibly resilient and very resistant to tensile forces. And when you look at it under light microscopy, because of this staggered array kind of thing, it actually has this sort of striated appearance. When you look at the light microscopic level, and this is important, this is the classic H&E hematoxylin eosin stain that you all messed around with in high school and then forgot about. The pink is the protein, the blue is the nucleic acid, right? And what you see are these collagen fibers, almost all of which are arranged parallel to the line of force in the tendon, but not all of them, right? There are also collagen fibers arranged in a crosshatch manner to give the tissue extra toughness, right? And you see the living elements, the tenocytes and tenoblasts, embedded within this collagen fiber matrix. But you see something else too if you look really closely. You'll see that, particularly over here, this image on your right, you can see that it's not really straight. There's what's called crimp in the collagen fibers. There's a certain amount of waviness in there, which gives the collagen fiber and the tendon a certain amount of give. And if you look at the physical properties of the tendon, if you look at a stress-strain curve, right, where the strain is the deformation that you impose upon the tendon and the stress is the resulting force within the tendon, you can see that as you begin to pull on the tendon here at the toe of the curve, right, most of the force goes into actually pulling those fibers straight. And so there's very little force response to the deformation of the tendon down here at the toe of the curve. But as you apply more force to the tendon and distract it further, you enter this linear physiological range, right, where the stress-strain relationship becomes linear. And this is where the tendon is designed to function most of the time, right? But as you apply more stress, you get into the elbow of the curve, the relationship becomes nonlinear, and you start to see microtrauma to the tendon. And this is the beginning of what we call tendinopathy. And then as you get stupid about stuff, right, and you get beyond a certain point, you'll start to see macroscopic failure of the tendon, and this will be full-blown tendinopathy. And of course, you can even get into rupture if the forces are high enough, or if the tendon tissue itself is pathologic enough. This slide, I don't want to belabor this too much. This simply shows you that tendon, right, undergoes changes as it gets closer to the bone and or muscle. It becomes more and more like the tissue that it's attaching to. So as you follow tendon towards its attachment to the bone, it becomes more bone-like in its composition. You go from tenocytes to mineralized fibrocartilage, you go through different cartilage types, and eventually you get this sort of like mineralized tissue that is almost indistinguishable from bone. So it's not like the tendon attaches to the bone, it's like the tendon slowly becomes the bone, right? Okay, so once again, to emphasize that tendons and ligaments are not passive force transmitting structures. They have viscoelastic properties that allow them to operate like springs, and CJ is going to talk about this more in his part of the presentation. They're more plastic and more trophic than cartilage, which means they can respond a little bit better to cartilage with loading and with training, but again, not with hypertrophy. Not really with hypertrophy. So you don't get hypertrophic ligaments. Stronger ligaments, if you look at the athletic ligament versus the untrained ligament, they're not that much bigger or thicker, right? They're just stiffer. Okay, so that's our quick introduction to the anatomy and the physiology of the synovial joint.